and to introduce my other colleagues to you. So it's my pleasure to introduce three extraordinary scholar scientists who will discuss genius in mind, brain, and molecules, respectively. I begin with Robert Michaels, one of the world's outstanding scholars in the psychoanalytic psychology of the mind. Bob is the Walsh McDermott University Professor of Medicine and University Professor of Psychiatry at Weill Medical College of Cornell. Bob served as chairman of the Department of Psychiatry at Cornell and psychiatrist-in-chief of New York Hospital from 1974 to 1991. Following his tenure as chair, he served as dean of Cornell Medical College and as provost for medical affairs at Cornell from 1991 to 1996. Bob is also a training analyst at the Columbia University Center for Psychoanalytic Training and Research and serves on the Board of Professional Standards of the American Psychoanalytic Association. Since 1970, he's been a fellow of the Hastings Center and a member of their board of directors. Bob is past president of the American Board of Psychiatry and Neurology, the American College of Psychiatrists, and the American Association of Chairmen of Departments of Psychiatry. He's a former member of the Board on Mental Health and Behavioral Medicine of the National Academy of Medicine. He's the joint editor-in-chief of the International Journal of Psychoanalysis, deputy editor of the American Journal of Psychiatry, and a former member of the editorial board of the New England Journal of Medicine and Psychoanalytic Quarterly. He's co-author of a classic text on psychoanalysis and the psychiatric interview, and has written important papers on the future of psychiatry. Please welcome Bob Michaels. Larry Swanson is the world's most important comparative anatomist of the brain. Larry is university professor and Appleton professor of biological science, neurology, and psychology at the University of Southern California. Larry studies how the nervous system controls thinking, feeling, and behavior. What are the design features of the biological circuitry? What are the parts? How does each one work, and how do they interact as a system? What is the wiring diagram? And particularly important for tonight, what are the historical foundations of this quest for the basic plan? Larry is a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Association for the Advancement of Science, and he served as president of the Carl Club and as president of the Society for Neuroscience, and is currently Secretary General of the International Brain Research Organization. He's recently authored a new book on neuroanatomical terminology. Please welcome Larry Swanson. Tom Jessel is the world's leading scientist studying development of neural circuits on the brain on the molecular level. Tom is the Claire Tao Professor in the Departments of Neuroscience and Biochemistry and Molecular Biophysics at Columbia. He's also an investigator of the Howard Hughes Medical Institute, director of the Zuckerman Mind-Brain Behavior Institute, co-director of the Kavli Institute for Brain Science, and co-author of the textbook Principles of Neuroscience. Tom's research examines the cell and molecular mechanisms that control the assembly and function of spinal motor systems. His work has defined how the diverse neuronal elements that form sensory motor circuits acquire their identity, how neuronal identity directs the formation of selected circuits, and how the precision of network wiring refines motor skills. Tom is a fellow of the Royal Society of London and of the UK Society of Medical Sciences. He's a foreign associate of the US National Academy of Sciences, a member of the National Academy of Medicine, a fellow of the American Academy of Arts and Sciences, an honorary fellow of the American Academy of Neurology. Tom has received several important recognitions, including the Kavli Prize in Neuroscience, the Gairdner Foundation Award, the Wilczek Prize in Biomedical Sciences, and the Gruber Foundation Neuroscience Prize. Please welcome Tom Jessel. Welcome to the night of genius in mind, brain, and molecules at the 92nd Street YMHA, in case you didn't know where you were. Ever since Socrates and Plato first speculated in the nature of the human mind, serious thinkers have sought to understand themselves and human behavior in general. For earlier generations, that quest was restricted to the intellectual framework of philosophy. In the 20th century, however, a school of philosophy concerned with the human mind merged with cognitive psychology, the science of the mind. Both then merged with neuroscience, the science of the brain, a merger that was energized by molecular biology. The result was a new molecular biological science of mind. The guiding principle of this new science is that mind is a set of processes carried out by the brain, an astonishingly complex computational device that constructs our perception of the external world, fixes our attention, and controls our action. Many people are now beginning to realize that a better understanding of the human mind in biological terms is the central challenge confronting science in the 21st century. The opportunity of a new science of mind opens up the possibility of using insights from this new science not only to understand ourselves better, but also to explore new linkages with philosophy, the social sciences, the humanities, and with studies of disorders of mind. Our purpose tonight is to consider the factors that contribute most seminally to the emergence of a new molecular biological science of mind, the people who would rank as geniuses in the area of mind, brain, and molecules. Now, there is no precise definition of genius, but I think most of us will agree that certain scientists, the three here and others such as Newton, Darwin, and Einstein, whose creativity led to seminal achievements that have revolutionized their work, are in a special class. All of these people have, through their work, permanently changed the way we perceive the world. This special class of individuals not only have exceptional intellectual ability, but more important, each of these people have extraordinary originality of the sort that leads to major new advances in their domain of knowledge. Persons with genius tend to have a strong intuition about their domains, and they build on these insights with energy. There often is no direct relationship between IQ and genius. For example, Richard Feynman, generally considered one of the most creative physicists of our time, had a rather unimpressive IQ of 125, and went on to win the Nobel Prize in physics and become widely known as a genius. You obviously need a minimal IQ of 125, but that is not sufficient. That IQ must be combined with personality characteristics such as drive, persistence, and creativity. Although it is hard to define genius, especially individuals of our own time, I would suggest that the key component of our definition be that genius is a person who's a game changer. Nonetheless, it is worthwhile taking a brief look at how this concept has evolved historically. In ancient Rome, a genius was a guiding spirit. The noun is related to the Latin verb genui, to bring into being, to create, to produce. Because the achievements of exceptional individuals seem to indicate the presence of a particularly powerful guiding spirit, by the time of Augustus, the word began to acquire its secondary meaning, that of inspiration, of talent. In the 18th century, Immanuel Kant defined it as the ability to understand intuitively concepts that would normally have to be learned by another person. Thus, for Kant, the essential character of genius was to be radically original. Kant writes, "Genius is a talent for producing something for which no determinate rule can be given, not a skill that can be learned by following some rule or other." In the 19th century, Arthur Schopenhauer added, "Talent hits a target no one else can hit. Genius hits a target no one else can see." This evening, we will attempt to apply the concept of genius to brain science and define who are the great geniuses in mind, brain, and molecules that led to making brain science in the 21st century the most exciting of all sciences, the sciences that teach us about our very nature. Who are the people that changed the game in each of these subfields of mind, brain, and molecules, and how did they do so? To accomplish this challenging task, we brought together three outstanding and original scholars. Bob Michaels will discuss genius in the study of mind, Larry will discuss genius in the study of brain, and Tom Jessel will discuss genius in the study of molecules of the brain important for mind. We will begin in turn. We start with Bob, then Larry, and then Tom, and ask each of our colleagues to end their discussion by spelling out for all of us what makes the individual or individuals they've selected a genius a genius. Bob, we begin with you.
Thank you, Eric. I'm going to talk about Sigmund Freud, and I'll try to underline several critical choice points in his work that point to him as a genius. As Eric has pointed out, the essence of genius is not intelligence, it's originality. A genius changes the way that people think. Intelligence can be helpful. It's essential for collecting supporting data, for planning observations or experiments, for integrating ideas from multiple domains. But that leads to progress, not to new pathways. Genius leads to new goals, new strategies, new ways of thinking. Behind me, you see 16 photographs of Freud, from a small boy to late in his life. It's appropriate to have that kind of slide for Freud because he, more than anyone else, taught us about the immense importance of infancy and childhood in shaping the character of the adult individual and his destiny. Here you see him with his father, and he's a good Jewish boy, with his beloved mother, whose love for him, he claimed, was one of the explanations for his optimism, self-confidence, and success in life. Freud was born in what is now Preborn, the Czech Republic, but was then Freiburg in the Galician region of the Austro-Hungarian Empire in 1856. His family moved to Vienna when he was a small child, and he spent most of his life there. He was a gifted student. After graduating from the gymnasium, received an MD from the University of Vienna in 1881. He did laboratory research on neuroanatomy, showed the similarity of the nerve cells of both lower and higher animals, conducted clinical studies of drugs, including some of the first studies of cocaine, and wrote books, monographs on aphasia and on cerebral palsy. Here you have him when he's 29 years old in 1885. The following year, at the age of 30, he left the traditional natural science laboratory investigation behind and began the private practice of medicine that continued for the rest of his life. This decision was importantly motivated by his need to make enough money to get married to his beloved fiancée. Freud's clinical practice was of outpatient neurology and psychiatry. He'd studied with the leading neurologists of the era, particularly Charcot in Paris, and his mentor in Vienna was the distinguished neurologist Joseph Breuer. Many of Freud's patients had physical symptoms for which at the time there was no explanation and no treatment. The leaders of the field assumed that these symptoms must reflect some yet unknown pathology of the nervous system. The patients were treated with sedation or hospitalization, but with no understanding of the cause of their problem or of any treatment that might be aimed at that cause. Freud changed all of that. He developed a theory of the mind, psychoanalysis, that postulated that our conscious rational thoughts are only the tip of an iceberg, that unconscious passions that stem from childhood define our character and shape and control our lives. Mental life, Freud pointed out, discovered, is lawful, not random, not chaotic. Understanding those laws opened the door to rational treatment for mental disorders. The hitherto misunderstood or ununderstood symptoms of psychiatric disorders were easily understood when Freud applied his technique to them of unlocking the secrets that lie behind his patients' free associations, dreams, fantasies, and the like. Freud's first dramatic clinical breakthrough was a simple one. Here at age 44, about 1900, at the time of his publication of probably his most famous work, The Interpretation of Dreams. I did something bad. Throughout the 19th century, his patients have been comforted, soothed, reassured, and ignored. Freud deviated from this standard routine. He did a startling thing. He listened to what they were saying, paid attention to it, and tried to understand it. He discovered meanings that had been concealed in their symptoms, meanings that seemed to be related to their problems. Further, Freud found that the patient's clinical course was affected by the process of making these hitherto hidden meanings explicit and sharing them with the patient. In brief, by turning away from speculation about unknown abnormalities of their brains and attending to the products of the patient's minds, Freud solved a problem that had baffled doctors for centuries. This is Freud on his only trip to the United States in 1909, accompanied by some famous colleagues, Carl Jung, Stanley Hall, the head of Clark University, in the middle in the front, and in the back, Abraham Brill, the American analyst, Ernest Jones, the British analyst, and Sándor Ferenczi, the Hungarian analyst. What were the meanings Freud was looking for? First, Freud traced them to adult patients' memories of abuse during childhood, largely of sexual abuse. However, he then discovered that these abuses had never actually occurred. The obvious next step for an honest investigator would be to correct this error, to publish a retraction. Instead, Freud had a far more important, a genius explanation. His patients had real memories, but they weren't memories of real events. They were memories of childhood fantasies. The study of psychiatric patients had led first to the role of mental life rather than brain abnormalities in the origin of their problems, and then to the importance of universal childhood fantasies rather than occasional episodes of child abuse as the stimulus for their mental preoccupations. However, the story's not yet over. Freud soon discovered that although he longed to be seen as an objective scientific observer who would cure his patients by sharing his discoveries with them, the patients saw him differently. They became emotionally involved with him. They craved his approval. They feared his disdain. The focus of treatment shifted once again, this time from the content of the patient's communications to the relationship between the patient and the therapist within which that communication occurs. The concept of transference became central. And to this day, almost all systematic study of the outcome of psychotherapy discloses that the most important variable determining that outcome is the nature of the relationship between the patient and the therapist. So we can trace a series of paradigm-shifting transformations in Freud's work, from simply caring for patients to listening to what they're saying, from finding out of stories of childhood trauma to recognizing that those are stories of childhood fantasies, from teaching the patient about their hitherto unknown thoughts which had become explicit because of the systematic investigation of their unconscious mental life, to examining the patterns of the relationship between the patient and the therapist, the patterns that repeat childhood patterns and that include the clues to the development of character and the development of symptoms. This slide shows Freud with his beloved daughter and disciple, Anna Freud. Freud is 57 at this point. It's in 1913. He's at the height of his scientific productivity. Freud, in fact, had made one earlier, important for us tonight, earlier attempt to understand his patients before the series of events I've described. It was a noble attempt, a brilliant attempt, but it was a total failure. He recognized this himself. He wrote an essay about it, but then decided not to publish that essay. Fortunately, it was preserved, and we have it available today. It marked his final foray into his neurobiology earlier in his career. It was an effort to explain the workings of the mind by developing a model of the brain that could explain thought processes. However, the neurosciences of the late 19th century were not up to that task. And he recognized that and turned his attention to psychological investigations of the products of the mind. He left the brain explanation of the mind, which he always believed would ultimately be where our field led to future generations. I'm privileged to be sitting among that future generation right here, right now. However, Freud never gave up the hope that someday the field of psychoanalysis would explain the mental products of brain functioning, and we could draw the links and bridges between those. This is Freud, age 65, toward the end of his life. Today, many of Freud's followers agree with his belief that a study of brain biology would ultimately provide the understanding of mental life and of psychoanalysis. However, probably today an even larger number of psychoanalysts believe that the shift of focus from the interior structure and activities of mental life in the individual's mind must include, for full understanding of psychoanalytic processes, must include an understanding of the patient's relationship with the therapist, and therefore go beyond the boundaries of the brain. Freud taught us to listen to our patients. He recognized that our mental life is determined and follows rules, just like the physical universe. He gave us insight into the fundamental role of bodily passions, of lust and aggression in shaping human behavior. And he taught us to study the developmental course of those passions, starting from earliest infancy. The evolution of his thinking is a story of repeated acts of creative genius. Thank you.
Wonderful, Bob. I have a slight disagreement with you. I agree with you, we're nowhere clear, nowhere near having a really profound understanding of the human psyche and the level necessary to deal with what Freud was dealing with, much less explain the outcome of psychoanalysis. But there's every reason to believe that ultimately one would get there. And I don't personally see why having a transference relationship makes it impossible for neuroscience to do it. We have relationship with our children, we have relationship with lots of people, but neurobiology is very much interested in what happens in the minds of people as they form relationships, as they break relationships. So as we get more mature, and we're far from being there, we should be able to do this. Maybe we can take this up in the discussion. One of the fascinating things about Freud is he was an outstanding anatomist. In fact, if you don't go out and buy it because you can't afford it, but if you ever pick up a copy of the current issue of Principles of Neuroscience, you'll see on the cover there is a ganglion of a fish that Freud had actually sketched out. He was the first person to show that Darwin was right, that the nerve cells of simple animals is very much like the nerve cells in your brain. He was an extraordinarily good anatomist, and I think would have continued in that if he didn't, as Bob pointed out, need the income to get married. But what is interesting is he was dealing with the same problem we're going to hear about from Larry, which is the critical insight into how the nervous system is structured. Larry.
Well, thank you, Eric. And I must say, I'm really delighted to be here. We have nothing like this in Los Angeles, so this is actually a real treat for me. This is the guy that I want to talk about. He's not a household name, Santiago Ramon y Cajal. He was a almost identical contemporary to Freud, actually, and with Pasteur and Darwin, but he's not a household name because it hasn't been possible to sort of encapsulate exactly what he did in a simple way. But I'll try and explain a little bit about why this guy turned out to be the most important scientist that was ever, the most important Spanish scientist in history. And he was certainly a peer of Darwin's and Pasteur's, but you can't say evolution or pasteurization in the same breath with Cajal. So I'll try in a few minutes to explain a little bit about what he did, which was basically to formulate several rules about how to think about the organization of the wiring diagram of the brain. And I think before I get into that, let me talk a little bit about him, because he's a little different than some of the other geniuses we've talked about. He was born in a tiny little, very poor village in the Pyrenees near the French border. So had a very, in a way, isolated upbringing. And he was a typical kid that liked to run around with his friends and get into mischief. And, but he loved to draw as a little kid. And his father put an end to that completely. His father was the local country doctor and would have absolutely nothing to do with the artistic talent of his son, which turned out in the end to be extremely important. But he was, as I'll get into a little bit more as we go along here, a real Renaissance man in many ways. When he was 16 out here in Aragon, taught himself photography, how to make negatives, built a camera, and took a series of self-portraits throughout his life, much like Rembrandt did. So here he took a picture of himself when he was 16. He had just read Robinson Crusoe and went out behind his house and posed like this. This is one of the last, this was 2 years before he died on the other side there, the Nobel Prize winner, who essentially revolutionized, he's considered actually the founder of modern neurobiology. So he actually, his father persuaded him eventually to go to medical school, and the way he did it was he got his son to help him in the dissecting room, making these classic drawings to help medical students understand the anatomy of the human body. And he got interested in medicine and went to medical school, got out of medical school and immediately got sent to Cuba, where he got malaria and came right back. And the consequence was that he and his father decided he actually didn't have the stamina to, for a medical practice out in the countryside. So he decided to become more of an academic research physician. Got a teaching job at the University of Valencia and was busy teaching histology, which is the microscopic analysis of the body, and was writing actually the first textbook of histology in Spanish. He was 35 years old, so mature, had a family, had 5 or 6 kids by then, and visited a friend of his in Madrid who had just come back from Paris with the latest methods in histology. And as he was writing his textbook, he realized that to him the most important unanswered question in neurology, and really the unanswered question of the 19th century in neurology, was how do individual nerve cells interact with each other within the brain, in the adult brain? What's the actual organization of nerve cells in the brain? And when he was 35 in Madrid, he looked down the microscope and he saw a slide that he looked at and said, "Eureka, I think I can answer that question." And it was a technique that had been developed about 15 years earlier in Italy by a man named Camila Golgi, who was also essentially a country doctor working in a backwater mental hospital somewhere in Italy, who stumbled across a reaction, a histological reaction that actually showed individual nerve cells rather than this incredible jumble. Before this method, it was like being out in the middle of the Amazonian rainforest, surrounded by incredibly dense thickets of trees and vines and everything else. With this method, it was more like being in an English garden where you could look at individual trees and plants and so on and really study them individually. And so Golgi invented this method, and he was looking down the microscope at his results and sort of went along with the current thinking in medical teaching that the interaction between nerve cells was a little bit like a spider web or looking at a fishing net from a distance. All of the cells seemed to be interrelated with no obvious organization. And it seemed like neural impulses would come into this system and go almost everywhere and somehow come out the other end. And when Golgi looked at his method, this is what he thought he saw, because it was the current thinking. Cajal wrote one of the great autobiographies in the life sciences. And he said as a kid, his most distinguishing characteristic was a disdain for authority. So when he looked down the microscope, he saw something completely different than the originator of this method. And I'll show you in this slide here, which is a very, he took advantage of this great skill at drawing to start seeing depicting what he saw through the microscope. And what you can see here up at the top is a human brain, and down at the bottom is the spinal cord. And if you see all those little things that look like trees with wires coming out of them and connecting, forming a circuit, it looks a little like Golgi maps, like Google Maps. He started to see actually how the circuit diagram of the brain is actually organized. And what you can see down at the bottom down there, coming out of the spinal cord, is a nerve cell with a little arrow going out to the left to a muscle cell. And this is called a motor neuron. This is how you move your finger or your leg, essentially, from your spinal cord. But what Cajal said was that the way this works when you actually think about moving that finger or that leg is that there are nerve cells up in the cortex of your brain that send an extension down to the spinal cord onto another cell that goes out to the muscle cell to move that muscle cell. And instead of being a reticulum or a spider web, the units of neural circuits are individual nerve cells. And they basically have three parts. When you look up at the top up there, you can see something that looks like a tree trunk with branches coming out of the top. Those are called dendrites. And they go into the sort of the main part of a nerve cell called the nerve cell body there, which then has a long string coming out, a wire called an axon. And this is the fundamental organization of nerve cells: dendrites, cell body, and axon. And these are the basic units of neural circuits called neurons. And this was completely different than the reticular theory that Golgi had held to. The second thing that Cajal realized was that nerve cells only let impulses go in one direction. And so information comes into the dendrites and goes down the axon. So by knowing what the individual units look like, you could start putting circuits together and predict the way that information flows through that circuit. So this was really done essentially in the 1890s. And with these two basic principles in mind, Cajal then spent about 15 years going through the entire nervous system of animals, for everything from rodents, humans, down into birds, amphibians, fish, and even invertebrates, and essentially came up with the basic way that we still 100 years later think about how the organization of nerve cells work. So he really revolutionized in a lasting way the way we think about the organization of neural circuits. And this is a very interesting diagram showing a basic wiring, part of the wiring diagram. So he was a polymath. As I mentioned, he wrote probably the second best biography in science after Darwin. He wrote a book called Life as Seen Through the Eyes of an Octogenarian, which is a classic of Spanish literature that's still in print. He wrote a book of cafe chatter of aphorisms. He wrote the first book on color photography. He was just an absolutely incredible person. And just to sort of wrap up in a big picture, Eric mentioned that neuroscience has a long history. I mean, it goes back to Hippocrates, Aristotle, and Galen, great thinkers in classical times. So there's about a 2,500-year history of neuroscience. And Cajal stands as one of the 4 or 5 greatest figures with lasting contributions in that 2,500-year period. He's one of the few scientists of the 19th century whose work is still referenced all the time. It's held up to scrutiny. He was a brilliant observer. And the final anecdote I want to tell you is that he and this man, Camila Golgi, shared the Nobel Prize in 1906, which I'm not sure this has ever happened before, but it was shared by 2 scientists who used exactly the same method, looked at exactly the same thing, and came up with opposite conclusions. And it actually took, most people believed Cajal, but it took until the 1950s for technology to get to the point where you could get definitive proof that Cajal turned out to be right. But he turned out to be right based on very precise observation and very clear thinking. So these were the basic attributes that I think set him apart, aside from incredibly hard work over probably a 40-year period.
Wonderful. Larry, what is so wonderful about Cajal is he specified for the first time that neurons interact at specific sites called synapses. Sherrington used that term. He was a contemporary of Cajal's. And that he suggested that probably what changes with learning is the strength of synapses. He had a vision that was really, decades ahead of his time. But what is equally remarkable is that Freud almost discovered this idea. And Cajal actually mentions him in History of Nervous System of Man and Vertebrates. He cites Freud's early suggestion that a neuron is an integrative actor, and it isn't just a diffuse syncytium. It's really quite fascinating. So, how does it all work, Tom? Put the chemicals into the picture.
So what I'd like to do is build on what we've heard already about the remarkable accomplishments and changes of mind brought about by Freud and by Cajal, and try and introduce the molecular nature of mind, brain, and thought. And this goes back to the early 20th century, where we're searching for the molecular mediators of brain communication. Your brain contains 86 billion neurons, each of which makes about 1,000 synaptic connections. So these synapses, which were inferred by Sherrington and by Cajal, are the nodes of communication. And so I want to introduce what a synapse really looks like for you. And so on the left-hand side here, we're looking at what is now a high-resolution image of the synapse. And you can see in the middle diagram there the presynaptic element, that is the synaptic terminal that is forming an association with the postsynaptic element, which is a dendrite in this case. And so Ramon y Cajal had showed that the neurons communicate at synapses, but the unresolved question was, how is communication actually mediated and affected? And you could think of 2 ways that this could, in principle, be achieved. By this time, we'd known, in the early 20th century, we'd known that the transmission of information from the cell body to that synaptic terminal is mediated by electricity, the propagation of an action potential. So one view is that electrical information transfers from one cell to the next in a form of transmission. But a second, radically different view is that the presynaptic element releases a chemical, and that chemical is released from the synaptic terminal and acts on the target cell. How could you resolve these differences? And this is what I want to introduce to you this evening. And so the 3 people who arguably made the greatest contributions to this issue are shown in this slide here. On the left is Otto Loewi, German-born, later became Austrian, and together with Henry Dale, a British pharmacologist, they worked out the nature and concept of neurotransmission that I will introduce to you. And the important thing is that you want to understand how that communication is achieved, both at the level of the releasing cell, whether it's electrical or chemical, and how that information is received by the target cell. And so John Langley, the third figure, also English, also trained in Cambridge, introduced the concept of receptor. And so the first crucial experiment in this series of studies was performed by Otto Loewi in an interesting way. Loewi, as early as 1903, had speculated that the mode of communication between one nerve cell and the next was chemical rather than electrical, but he'd failed to come up with any way of testing this idea. And so it wasn't until 1920 when he, just before Easter, on April the 3rd, went to bed that night, and he had a dream, and it was a remarkable dream, because he woke up in the middle of this dream and realized that this dream was telling him how to perform an experiment to test the idea that he'd had 17 years earlier. So he scribbled down notes, went back to sleep, woke up in the morning, and found to his exasperation that he couldn't read his own notes. So he'd lost the ability to do this critical experiment. So depressed, he went to bed the next night, the night before Easter Sunday, April the 4th, and the dream returned. And this time, in his prepared mind, he managed to write in copperplate script the nature of this experiment. He couldn't wait. He couldn't go back to sleep. He got up 3 o'clock in the morning, went off to the laboratory, did the experiment that proved that transmission was chemical. This is the real stuff of science. Eric Kandel wakes up in the middle of the night, most nights, goes off to the lab, does an experiment, phones you up to tell you about it. This is the vibrancy of doing neural science visited. So what was this great experiment that Loewi dreamed about? This is the idea that he dreamt this stuff. It's the stuff of dreams. This is Loewi's description of the idea of the experiment, the immediacy of going to the laboratory in his own words, and this was the experiment. So on the left-hand side, you're looking at a heart that has been taken out of a frog, and the vagus nerve that innervates the heart, and this was all known. If you stimulate that nerve, as shown in the lower traces there, the rate of heart beating slows dramatically. This was a known fact. What the dream told Loewi to do was that if this was chemical, you should be able to take the medium which was bathing that heart and apply it to a second unstimulated heart. And if it was chemical rather than electrical, there was a chance that that second heart would show the same response as the first stimulated heart. So Loewi hooked up these two hearts, he did the experiment, and lo and behold, the second heart slowed as the first had done. So this, if you like, was definitive proof that the transmission stimulated by vagal nerve activation was chemical rather than electrical in nature. So you might have imagined that the world would flock to this experiment and accept it in, dramatic terms. Not a bit of it. The skeptics of the science world took a decade to believe Loewi's experiments. And part of the reason that, scientists are trained to be skeptics, to be dubious of novel ideas. And part of the act of genius is to overturn that nascent skepticism. And one of the reasons that they gradually began to believe in Loewi's dreamlike experiment was the work of Henry Dale. And Dale and Luries were close friends. They had interacted. Luries had come to England to work in Starling's laboratory. And Dale's insight was to define the chemical nature of the neurotransmitter that Luries had inferred, this Wegestoff. And so what Dale showed is that a small molecule termed acetylcholine, on the basis of its chemical structure, was the mediator that achieved this slowing of the heart upon vagal nerve stimulation. And Dale went on to show much more broadly that acetylcholine is a major transmitter in many different synaptic communications. So the motor neurons that Larry showed you that innervate muscle also use acetylcholine. And this was demonstrated by Dale. And together, Dale and Luries gradually overturned the skeptical view of chemical transmission to the point that in 1936, those two were awarded the Nobel Prize in Physiology and Medicine. Now, the third member of this group, John Langley, had assumed that if there are chemical transmitters, they have to act on some specific element on the target cell, what he termed a receptor, and that those receptors must be molecular entities just as the transmitter is a molecular entity. And now we know that Langley is true, and some 80 years after Langley's insights here, we have an enormous information trove on the function of these receptors. So on the left-hand side, you're looking at two receptors for acetylcholine at the nerve-muscle junction. We know that those receptors open in response to the binding of acetylcholine. They let ions through that then activate the target cell. And we even have molecular information. So the right-hand picture, that blue and yellow picture, is actually an atomic scale resolution of the receptors for acetylcholine that Henry Dale had demonstrated to be functional at neuromuscular junction and in that vagus preparation. So through unlikely sources, the presence of a dream, we have come to understand the nature of chemical mediators. And the power of dreams is illustrated by a second example, actually an earlier example, where the German chemist Kekule had been dreaming of how benzene, a structure that we now know and take for granted, could occur. And he dreamed that there were little atoms that in a sense chased their tail, that is the image on the left, to the point that this dream gave Kekule the inspiration to think about the aromatic, cyclic, circular nature of the benzene molecule. And that itself has implications for neurotransmission, because we're looking at another neurotransmitter on the right-hand side, which is called dopamine, which is, you can see, has side chains off this core benzene moiety. And so dopamine turns out to be a transmitter that gets us back in direct way to the idea that disease process and even the biology of thought are dependent on this transmitter. So if we look in this next slide here, the loss of dopamine is associated with various neurological disorders like Parkinson's disease. So on these images shown on the right, the one with the red box around it is a normal marker of dopamine stained in an individual who died of some other cause. And all of the other images there show the reduced levels of dopamine with increasing time of the diagnosis of Parkinson's disease. So I think you can see 1 year, 2 years, 4 years, gradually. And what you can see is that by 3 or 4 years, there's virtually no dopamine marker in these small neurons in the brainstem that provide the source of dopamine. So this is an indication that transmitters and dreams have a reality in terms of the neurology of today. And also, the dopamine in a different neural pathway is relevant to disorders like schizophrenia, disorders of thought. So here is dopamine transmission and the biology of thought. So what you can see is that changing the levels of dopamine can produce the symptoms of schizophrenia. Increasing dopamine produces schizophrenic symptoms, and drugs that inhibit the actions of dopamine alleviate schizophrenic symptoms by neurons that we know are different from the neurons that mediate Parkinson's disease. So here we've gone from the nature of chemical transmission, its initial demonstration, to the idea that many aspects of movement disorders, neurological degenerative disease, and even disorders of thought are dependent on these transmitters. And we now think that there's some 30 to 100 different transmitter substances used by the human brain. So a vast diversity here, even though probably a dozen of them so do 90% of the work. So transmitters are important. The fact that you're thinking about what we've been talking about is a consequence of transmitters that are going on in your brain. And I just want to finish by pointing out that even the greatest literary minds of our time, E.M. Forster, arguably the greatest stylist, intuitively had the sense to understand about neural communication. Because in his book, Howard's End, E.M. Forster wrote this little introductory, only connect. So if we knew, and people are still working on the only connections that exist in the brain to understand degenerative disease, thought, and just the basic discovery of communication, which is, I would argue, the work of these three geniuses.
That was simply wonderful. So when we return here 40 years from now, I think we'll be in a position to evaluate how far we've come to meet your challenge. Can we ultimately get to the point where we can explain in biological terms many of the aspects that Freud was talking about? And I, just to open up the discussion, just want to raise a few of these issues. First of all, there's been detailed studies, both from cognitive psychological point of view and from imaging point of view, of unconscious mental processes and how information moves from unconscious to conscious. And we're beginning to see how that occurs, number one. Number two, we see in terms of the chemicals that are involved that many of the disorders of the brain, including those involved in thinking and acting, involve critical chemical transmitter systems. But finally, I think one of the most fascinating things is that a series of experiments that Helen Mayberg did. Helen Mayberg found that in people who were depressed, she was interested in imaging people who were depressed, there's a characteristic change in activity in an area of the brain called the anterior cingulate. She then exposed these patients, she divided them into two groups. She gave one selective serotonin uptake inhibitors, the conventional pharmacological treatment for this, and she gave the other group psychotherapy, cognitive behavior therapy, Ehrenbeck's treatment. And she found that psychotherapy worked exactly as well, in fact, many cases better than the antidepressant medication, not only in the effectiveness, which had been known before, but it produced an anatomical change. It reverted the change back to normal. So psychotherapy is a biological treatment. It works through neuronal processes, it works through synaptic connections, and it uses the chemicals we talked about. So hopefully when we come back next time, we'll be able to give you a more complete explanation. Let's go around one quick round and finish it up. Bob, you begin. Have we convinced you?
I don't think we'll be back in 40 years, Eric.
I think you're right.
You haven't convinced me of that. There's no question that mental life is a psychologically biological phenomenon. There's nothing else it can be. There is no mental life without a brain. There's no way that people can change without changes in their brain. However, that doesn't mean that, at least in the foreseeable future, what we know about the brain is going to give us an opportunity to understand the fine texture of the mechanisms of psychological processes. We can tell that unconscious mental life can be transferred to consciousness and know something about the neural pathways involved in that. But we can't tell very much about the content of the ideas involved in that by studying the brain. We can, you heard about some critical dreams in the history of neuroscience. We know a lot about dreaming and the neuroscientific basis of dreaming, but we know almost nothing about the neurobiology of specific dream images, of the circle of snakes that Kekulé used in discovering the benzene ring, or the images that are involved in every one of our dreams every night. Will we ever be able to learn enough neuroscience so that we can translate all of the psychological knowledge that we can develop into neurobiologic knowledge? That's a very exciting and interesting question, and I think that goal will organize a huge amount of interesting work in the next century.
Tom.
I want to hear what the questions are that you've got sequestered on your lap, Eric.
These are not my questions. These are other people's questions.
I know, that's what I want to, want to.
Do you just want to make one comment, Larry, one comment before I go to the questions?
I think the characteristics of genius are to look and see what other people have ignored. And in many cases, Cajal saw what Golgi didn't see, Loewe dreamed, imagined what, and so this is what genius is all about. And when you think today, the information load that is available in some ways is a dampener on the freedom of thought. We have too much information about how the brain works and functions, and so it's important to step back from that, I think, a little bit, and try and think, you know, ab initio about the questions that you're addressing.
One final word from me, Larry, before I go on.
Well, maybe two final words.
Please.
I
meant metaphorically.
Eric has, said that the great success of 20th century biology was the decoding of DNA, and that probably the 21st century will be decoding the brain, which I think there's a lot of truth to that. Tom explained brilliantly, I think, that in the 20th century we really figured out how individual neurons work beautifully. What we're missing now is something called systems neuroscience, basically a theoretical framework where you could actually think about mind, for example, rather than individual neurons. And the one breakthrough in thinking here is this human genome project, which essentially decoded our DNA. And it was a tremendous undertaking to actually sequence 3 billion letters of DNA, basically, in the human chromosomes. It took a lot of time, and a lot of people now think that, and that led to incredible breakthroughs, actually, in medicine and life sciences, thinking about the big picture of heredity. People are now thinking that a complementary thing in the 21st century is the wiring diagram of the brain, basically how it's organized, so that we can actually begin to think about the more complex types of behavior. And the problem really is that it's very hard to recognize genius until you've seen it. So chemistry, probably most people have taken chemistry, and the first thing you see on the blackboard is the periodic table of the elements. I mean, it's a beautiful way of organizing your thinking about chemistry. Watson and Crick gave us a double helix model of DNA, which was the theoretical framework for basically genomics. What we're really missing so far is a theoretical framework for thinking about the organization of the brain. So I think we're waiting basically for an Einstein or a Darwin or somebody to come along and in some way come up with something that really has lasting value, which we don't have at this point.
Do you think genius is something you're born with, or can it be developed?
What I would say is that the nascent capacity for genius is probably there very early on, but that what happens is the vagaries of life either permit that talent to flourish or suppress it in different ways. And so, yes, it's there. You have to have this innate capacity to think differently. But too many things happen along the way
Absolutely.
that will filter that out.
And this is why we love to read obituaries, for example, because every extremely successful person has a different story. They get there in a different way, and it's very hard to figure out any general rules about. On the other hand, a Mozart, someone with that unbelievable musical talent at an early age, it's hard to imagine that that wasn't
You see, Cajal
inborn
in some
somehow.
ways is more remarkable than Mozart, because Mozart's father was a very talented musician. He was embedded in this. Now, granted, Cajal's father also knew something about medicine, but nothing in the league that Cajal did. Cajal really, not only that he came in Spain, which was at that point rather unintellectual environment, and he was really just a genius in the way he stood out.
I don't think there's any answer to it, but I think, almost
There are probably a number of different avenues clearly that lead to this.
everything in biology is a bell-shaped curve pretty much, and I think you're going to have way out on one end some unbelievably rare combination of factors.
Yes. Have you considered the possibility that consciousness, awareness, memory exist externally to the brain and are localized and received by the brain rather than generated, similar to the relationship of wireless laptop to a server? Why don't you answer that one?
I can give a very simple answer, no.
Good. Absolutely. Everything comes from the brain. There is nothing else. This is a difficult question.
This is an assumption, Eric, and it's an assumption that is an essential assumption. I think all of us, we think very differently, but the thing that binds us together is the thought that the basic laws of chemistry and physics, even though we don't understand them at the moment, are going to apply to brain structure and function if we only were.
I think that when Penfield came along and in unanesthetized patients could stimulate different parts of the brain, particularly stimulate the hippocampus and elicit memories, what else do you want, baby?
I want the circuitry that underlies that.
Well, that
molecular
took a lot. It's
mechanism.
coming along. How is the brain of a transgender person different from somebody who is cisgender?
There's actually no, I don't think, any good biological data on that.
But there are people who've addressed this in the context of variant behavioral traits. Simon LeVay, who was a very distinguished visual system anatomist, began to get intrigued by this. And he demonstrated, as other people, Bob Hammer, I think, has demonstrated there are structural differences in the brain that go along with gender and sexual predispositions that we don't understand. We don't understand most things about the brain, despite your presence, Eric. So
I never deny that. I said we have to come back a few years later. You're absolutely right.
Eric, more in keeping with the topic of the evening, we certainly can't tell the difference between the brain of a genius and the brain of someone of normal intelligence. We don't yet know enough to be able to
I
differentiate.
think that's probably true, but there hasn't been a systematic examination of that question. But you're right. I'm not sure we'd pick it up. Also because it's quite likely that genius is not a monolithic concept.
Certainly.
I think genius is a very complicated term to apply to the pursuit of science, actually, because there are many people who've made fundamentally important contributions who you would not think of as geniuses. They're simply progressive, iterative, determined workers on the field, and those contributions can be just as illuminating as the work of an unbridled genius.
I think, and this is the note I think we should end on, well, you can example. What really strikes me is that when I entered the field in 1957, when I took an elective, actually when I went to the NIH and spent 3 years there, it was amazing how little we knew about the brain. And just during my lifespan, the progress has been enormous, and it's accelerating. So I think we have every reason to believe. I mean, who would have thought? We now feel, many of us are confident, with the next 5 or 10 years we'll have not only good insight into Alzheimer's, which many of us think already exists, but we'll have useful treatment for that. That would have been inconceivable 15, 20 years ago. So I think the progress that is coming along is really very encouraging. On that note, unless somebody has anything else, I want to really thank these three guys. They're really terrific.
Thanks.